Get started in just a minute. Hey, Alden. Looks like we've got Alden Hawkins on the line.
Hey, Alden.
Thanks for joining us. We're going to at least try to stump the herpetologist today. We'll see. Welcome to our live stream of Stump the Herpetologist. We've got Paul-Erik Bakland. This is Mark McKnight on the line from Reflection Writing and Hamilton County Department of Education, I guess, at this point. Thanks for joining, Paul-Erik.
Yeah, thanks for having me. Glad to be here.
We would love if you, I see we're starting to get a few people logged in. If you just logged in, give us a comment. Let us know where you're joining from. Say hello. We have one so far. We have Alden that says hello to everybody. We'll get started in just a second. All right, I'm going to kick us off. Hey, if you find this interesting, please subscribe to our YouTube channel. Follow us on Facebook. You can also find us on Instagram at Reflection Writing. Thanks so much for joining today. Like I said, I'm calling this Stump the Herpetologist with Paul-Erik. We'll see if we can stump him or not. We'll begin in just a minute. But first, I want to say one of the things that we're trying to do out here is support wildlife, and we are encouraging you to do the same thing. And one of the best ways you can do it is to give them something to eat. This is a prothonotary warbler that I caught during the City Nature Challenge, and actually I got to see a different one, but a little bit further down the creek this morning, I got to see one. This is a beautiful bird that comes back to the Chattanooga region to nest here, and hopefully, if they find enough to eat, make some more prothonotary warblers. This is an eastern phoebe, and as you can see, it has some kind of beetle fly thing in its mouth. Haven't identified that just yet, but these birds need something to eat, and of course, for them to have bugs to eat, they're going to have plants. So one of the things that you can do to help kind of restore the natural balance here in the Chattanooga area is to plant native plants. Shop online. We took this is what the native plant sale would have looked like and normally looks like in our 34th year of doing it every spring. But this time you're gonna have to go online. It's not quite as fun, but still have lots of great plants for you at reflectionwriting.org/plants. There's a plant list there, and then at the top of that list there is a link that you can click, and when you click that link, you'll see the. I just heard that it's just showing black on YouTube. Not sure why. Looks like we're on Facebook, but might be having an issue on YouTube here today. But at any rate, we will consult with you guys. If you don't know what you want, you can actually set up a time to have a call with our plant guys, and they'll help you choose what you want. So again, this guy is calling out for you to do that. This is the one of the last webinars that we're doing in our series about the City Nature Challenge. The City Nature Challenge is organized globally by the California Academy of Sciences and the Natural History Museum of Los Angeles County. All right, sorry guys. Sounds like Alden can't see the image either, so it must be something with my screen. I'm actually going to switch. All right. Hey, Alden, can you see this now? Can you see the slide that says City Nature Challenge? Okay, cool. Looks like we can see it now. So let me actually go back. This is annoying. I was actually just telling Paul-Erik before we logged in that apparently I need a third screen to be able to do this right. Anyways, Stump the Herpetologist with Paul-Erik Bakland. Here's the prothonotary warbler that you apparently could not see a second ago. Beautiful little yellow bird, and it's very loud too. So once you hear the call, you'll know it. This is the insect that that eastern phoebe is eating, and that birds need to be able to procreate. here is what the native plant cell should look like. Go check us out, reflectionwriting.org/plants. And then, like I said, this is one of the final webinars that we're doing in the City Nature Challenge 2020. The observation period was from April the 24th to the 27th, and we are currently in the identification phase. And so we thought it'd be fun to have Paul-Erik come talk to us about identifying amphibians and reptiles. And that ID phase ends on May the 3rd. So we have through the weekend to do this, which is great because I actually haven't even finished uploading all of my observations. If you have any observations that you haven't uploaded, get them in as soon as you can, because obviously it may take some time to identify them as well. If you haven't signed up for iNaturalist yet, go do it. iNaturalist.org, download the iOS app. It really is a lot of fun. I've met a ton of people and have learned a lot. It's kind of like nature flashcards. Here's how it works. You observe the community. You can identify. iNaturalist has a really good AI algorithm that will help try to identify for you. There's a discussion that can happen, and experts from all over the world come in and say what they think it is. And then ultimately, when you get to research grade, it is sent up to a global database that scientists can use to study ranges and all kinds of interesting things about what people are observing. Please, when you're signing up, leave this box checked that says that you license your photos, sounds, and observations so that scientists can use your data. This is what makes it useful, is if you allow this to happen. So what we're trying to do with this whole identification thing is first off, we're trying to get you guys to observe, make observations that are verifiable. And then the other half of that is that the community needs to come in and agree on an identification, and then it becomes research grade. To be verifiable, we need a date, we need a place, we need a photo or a sound, and then it needs to not be a captive or cultivated plant or animal. We don't really care if there is a tiger in Chattanooga, if that tiger is at a zoo. And then one of the big things about this, the photos and sound things, for the photos, we need enough visual information for others to help with the ID. If you haven't already seen the episode that we did about getting good photos for plants, that's an interesting one to try out and go check it out. So without further ado, I want to introduce Paul-Erik. And Paul-Erik is a longtime volunteer here at Reflection Writing and has been a part of what we're doing out here for about 7 years. He actually began volunteering in his last year of undergrad at UTC, where he was majoring in biology, and he was assisting research in Dr. Wilson's herpetology lab, which is called Team Salamander. Following that degree, he was accepted as a Master's of Environmental Science at UTC, where he kept working with the herpetology lab. And he'll tell us a little bit about his thesis research, because I can't pronounce it. But after his master's thesis, which he actually presented and defended here at Reflection Writing, which was fun to watch, although I didn't understand half of what he was talking about. And he was hired as an adjunct faculty at UTC in the biology, geology, and environmental science department, and was working there as an ecology lecturer and lab, and then has now moved into the Hamilton County school system, which is awesome for us because we work really closely with Hamilton County schools. And to Paul-Erik, you're now there teaching biology at East Hamilton High, is that right?
Yeah, that's correct. And actually, last semester I got to teach kind of like a pilot test or trial run of an intro to herpetology class for high school students, and that was kind of exciting. That's something that I'm hoping to develop down the road. I'm not really aware of high school herpetology being offered elsewhere, so hopefully that's kind of a unique thing that can help set us apart and be interesting and engaging for the students.
Awesome. So let's go ahead and get something out of the way to start. What is herpetology?
Well, herpetology is the study of reptiles and amphibians. And from an evolutionary standpoint, it doesn't make a ton of sense that those two groups are lumped together and studied together, because they're not each other's closest relatives. But they've always been studied together that way for a few different reasons. It's largely that they can be sampled in many of the same ways. So rolling a log in the forest, you have almost equal chances of finding a ringneck snake as you do a slimy salamander. So you can survey for them in many of the same ways. And so yeah, herpetology is the study of reptiles and amphibians, and it's very broad and encompassing of many different like subsets of that type of research.
So it's kind of a study of the stuff that slithers around in the forest.
Yeah. So the study of creepy crawlies, comes from the I believe it's Greek word herpeton, which means thing that crawls. So yeah.
All right, I like it. All right. Well, speaking, just personally, this is what I get a lot of times when I'm out observing things right here. So, if you come out to reflection routing when we're open again, you can pretty much catch a turtle, most of the year if you go along our creek bank around Lookout Creek or on the boardwalk. But to me, and a lot of times it's just the contrast and the light, they all kind of look the same. And so one of the things that I've found that you can do once you get back home, at least, is zoom way in on that photo and then give it some contrast, just blow out the, it's not a beautiful photo necessarily, but you can start to see some identifying characteristics. So Paul-Erik, can you talk me through what we're looking at here? First off, is that recognizable to you? Probably is anyways.
Well, I'm glad that you've called this Stump the Herpetologist, because that's probably pretty accurate when it comes to turtles for me. Unfortunately, that's probably my weakest area, but I'll do my best here. So what I believe we're looking at here is a slider turtle. So we actually have pretty good diversity of turtles in Southeast Tennessee, and we actually have a couple of subspecies of slider turtles. So we have the common pond slider, which is Trachemys scripta, and then we also have the Cumberland slider, which is Trachemys scripta trucii, and I think that's what we're looking at here. So the identifying characteristics there is on just the common pond slider is there's yellow blotches behind the eye, whereas on the Cumberland slider they tend to have these narrow yellow lines running down the back of their eye, and they also have narrower lines on the legs as well, and you can't really see that well in this photo, but if I had to bet, I would say that this is probably a Cumberland slider. It's for sure within Trachemys scripta. It's a subspecies of Trachemys scripta.
Cool. Very good. One thing I kind of jumped right past, and I got too excited about the ID section here, but tell me a little bit and tell the viewers here about your thesis research and what exactly you were working on there.
Sure. So I was studying an infectious disease of amphibians. It's a ubiquitous thing all over. It's on every continent that has amphibians, and it's causing widespread extinction and extirpation events all over the world. It's called just kind of commonly chytrid fungus. So it's a disease that's caused by a fungus. The Latin binomial for the fungus is Batrachochytrium dendrobatidis, or just BD. So it's called a few different things, either BD or chytrid fungus, but it causes a disease called chytridiomycosis that often results in death of an infected individual. But what they've found is that not all species are impacted equally by it. So it comes down to a variety of factors, but one of the major ones is some physiological things. So the keratin content in the skin seems to have an impact on how adversely affected different species are. But there's also some ecological effects as far as prevalence of the disease is concerned. And so that's where my research was focused, was kind of investigating these ecological differences to kind of assess what things impact the prevalence of this disease. So it is documented in Tennessee. We've got it in Tennessee. It's all over in the United States, essentially. And so what I was doing was evaluating the role of habitat type specifically. So I sampled frogs all across East Tennessee at wetlands and at urban retention ponds to determine if there was a difference between infected individuals at retention ponds and wetlands. And unfortunately, the results were somewhat inconclusive. While there was a numerical difference, it didn't turn out to be statistically significant. So I also used watershed basins to kind of separate my study sites. So I had like a northern watershed basin and a southern watershed basin. And kind of weirdly enough, all of my positive samples for this fungus in the southern watershed came from wetlands, while all of my positive samples in the northern watershed came from retention ponds. And so, as is common in science, this study raised more questions than it answered. So that's exciting on one hand, but a little disappointing that we didn't get a little more clarity on the other. So it's an interesting thing. There may be some ecological differences going on there, but for the time being, it appears that wetlands and retention ponds, at least what the numbers are saying, have equal likelihood of having positive individuals.
Interesting. Just want to let everybody know who's watching. If you have questions, drop a comment in on Facebook or YouTube, and I'll pose the question to Eric. I'm going to switch over real quick to our City Nature Challenge project, and I'm going to walk through a couple of things that we see over there. One of the things that's fascinating to me about this, this platform in general, but this project and the City Nature Challenge, which is a global challenge, it's just interesting to see what people find and to see the stats section down here. So if you click through on the stats, we can get more detail. You'll find this, which shows, first of all, the number of observations that still need an ID. And you see on here, we're at about 54% that still need an ID. So we need your help. If you have any specific expertise, or if you're just a naturalist in general and you can pick off some of the things that you recognize, please do that for us. We have about 41% that are research grade, and like I said, going up into the various databases where scientists can use this data. And then we have a couple hundred casual observations. Casual observation is something that is either captive or cultivated, like I was mentioning. It's not that there's no value to those, but they're just not useful to scientists in terms of studying range or anything like that. Some of those casual observations are mine. There are some legitimate reasons to put a casual observation in. For example, I kind of have just like a frequency thing that I'm keeping track of, where when we walk the dogs at night, I often see a cottontail rabbit, and I'm not going to get a picture of it. It's nighttime. It's moving too fast. I don't have my phone out anyways, and I don't care. I'm just recording when I see it because I'm curious as the south side of Chattanooga continues to develop. Am I going to start seeing more because they're being displaced and they're running all over our yards, or will we see fewer? Who knows? But I just think it's kind of fun to pay attention to what's going on around us and to record that information. The other thing about a casual observation, we do have a lot of animals in captivity here at Reflection Riding, and especially with the, I always think about the eastern screech owl. I've heard eastern screech owls. I've still never seen one in the wild that is close enough that I can actually take a picture of it. But here, if you come visit when we're open again, you can actually get right up close to that screech owl. And so taking that picture and uploading it to iNaturalist actually adds to the algorithm that helps identify things visually. So there is a little bit of actually usefulness to doing that. But here's where it gets really interesting. This is actually broken down by, what is this, kingdom? This is kingdom, right? I'm not a biologist. Plants, that's a kingdom. That's a phylum. That's King Philip. That's the Philip part. Okay, so phylum, so we have plants. We have the vast majority of the species here, 60% are plants. That's no surprise. They're kind of the baseline for biodiversity. We have 12 mollusks. We have 16% are insects, 169 species. And then 29 arachnids. And then you get over to the creepy crawlers. We got amphibians. We have 16 species of amphibians and 14 species of reptiles that are identified so far. Paul-Erik, based on what you know about the biodiversity of the Chattanooga region, does that sound about right? How do we do in terms of finding the herps.
Let's see. Okay, so say that again. So reptiles, how many reptile species?
14 species.
Okay, and
then
amphibians?
16 of amphibians.
Okay, yeah, that's pretty good. There's still a lot more to be found, but obviously, you know, habitat on a smaller scale, you know, doesn't necessarily, like what you guys have at Reflection Riding, maybe is not suitable for everything that could potentially exist in the region. So I'd say, you know, that's pretty good for this area for sure. Yeah.
It's also a little early in the year. It was chillier than we were expecting.
Mm-hmm.
We've certainly had some late April bio blitzes where it's been pretty hot, but it was relatively chilly too. So, all right, let's see what we have. One of the things you can do is switch over. I just clicked on one of those two slices. So if you click on each of these that pops up, you can actually click on it and it'll open a new tab. And so you can see specifically what people are finding. And you can actually use these filters. So this is all that did when I clicked on amphibians here. It just created a page here of observations that's then filtered by amphibians. So if I get rid of this, we just go back to all the observations in that project. So we have 3,300 observations. So I'm going to hit back and just do the amphibian. But that's just something to know. If you're looking for something in specifically, you can type in anything into this. All right, so we have a decent number of frogs that people found, toads, salamanders. This cave salamander actually is super cool. I've called this one out in one of my little blog posts during the challenge. But this is Betsy Bowden that found this cute little cave salamander, and she got a great shot of it too. Give that a second to load here. So,
Mark, I need to make a correction to what I said. That was broken down by class, not phylum. My mistake.
Ah, okay. Well, I didn't see anybody calling you out in the comments yet, but I'm sure somebody would. Sure somebody would. All right, so let's see if we can find something that has not been identified. I'm going to go back to what I was just looking at a second ago. And then I'm going to add one more filter, and this is a filter that's useful if you are going to try to help us with the IDs, which I know you're going to if you're watching this and you're interested. So go down to a taxon that makes sense for you, and then click onto the filters, and then click this one under Show that says Needs ID. And then you can click Update, although you probably noticed it just updated behind the scenes anyways. So all that update does is just close. All right, so these are species, but we want to switch back to individual observations. All right, let's see what we got. American toad. All right, now this one always gets me because there are two Anaxarus. Is that how you say it?
Anaxarus. Mm-hmm.
There are two Anaxarus in this region, and I can't tell the difference. Tell me what I should be looking for here.
Okay, so there's a couple of things. I think that the easiest way to identify these guys is through the number of warts per spot. So you see these dark spots on the back, and if there is 3 or more spots in essentially any of the or 3 or more warts in any of the spots, it's likely that it's a Fowler's toad. And so American toads have 1 or 2 spots, or 1 or 2 warts per spot, whereas Fowler's toads can have spots that contain three or more. There's also a proximity of the cranial crest to the eye that is used to identify where it's maybe confusing, because like in this individual, I guess there is a couple of spots that have 3 warts. So I would go with Fowler's toad on that one. So yeah, I think that's probably the easiest characteristic to use, the number of warts per dark spot.
Okay, now I wouldn't usually do this because like I said, I don't know, so I'm not going to identify things I don't know. But because I'm talking to Paul-Erik and I believe him, what I would do to make that determination is I go in and I click suggest an identification, click the species name, and then what I would really do is I would just do this one, which doesn't really help at all. But you'll see on here actually that the first suggestion is Fowler's toad. So the AI has actually already made that that same determination that visually this to them looks like a Fowler's toad. You'll also see that there's one that we don't have here. The Great Plains toad doesn't occur here. However, according to the AI, it's the second most sort of similar from the algorithm. And then third is American toad. So it's totally possible that the algorithm, I mean, there are plenty of correct IDs that show up as the third or the fourth option here. So don't, you know, the AI is only so good, but in this case, it looks like it's figured it out. Now, one of the things you could do if you wanted to go research more is you can click this view button, and it'll actually take you over to the species page, and you can learn more about that species. But in this case, I'm just gonna go ahead and pick that one, and I'm gonna say for Paul-Erik Bakland, because I don't actually, I'm not claiming that. And then I'll hit suggest. Now, what's gonna happen is it's gonna change up top. It's gonna change it to genus and Axeris because we have conflicting IDs here, and it shows that I disagree that this is an American toad. Now, if we have a third person come in and ID and they agree with one of the two of us, it'll switch back to one of the two. And then you see this community taxon right here. What this is telling you is how many people think it's one thing or another thing. In this case, more than 2/3 believe it's something in this genus. Eventually, as we get more IDs, it'll be more than 2/3 believe it's one or the other species. The other thing you can do is annotate. So I'm going to go ahead and say this is an adult. Do you agree with that, Paul-Erik?
Yep.
And I'm going to say that it's alive, seems to be alive. And then this is data that will, well, it's an annotation. So basically it rides alongside this observation. And so if I wanted to go, if I were studying, let's say I were studying North American toad mortality, I could actually go look at the map, narrow down to the ones that were marked as dead, and then start digging in and getting more detail. And that's the kind of thing that people will do on this platform, and is really fun.
You know, Mark, if I might, I'd like to add something to this. I think that,
yeah.
You know, what's so cool about iNaturalist is it has essentially what is a built-in peer review function where, you know, you have to have a certain number of people saying it's the same thing for it to go to research grade. And that's wonderful. And just because something goes to research grade doesn't mean it's permanently there, because it's possible that 2 or 3 people incorrectly agree on an identification, and then somebody else comes along and says, no, it's actually this. Then it goes out of research grade, right? Back down.
Absolutely. Yeah.
It's a continuous debate, and that's great because that's exactly what we want in science. So that's awesome that iNaturalist is something that can involve the public, but also maintain some of this, you know, these structures that make this robust.
Absolutely. And it really is fun to be a part of that science. And I've been on the platform long enough now that I've actually, I've seen it firsthand. I've had a researcher from the USDA reach out to me and say, hey, look, you have the southernmost observation of this plant that I'm doing a management plan to protect. And can you tell me, because I had obscured the location because it's an endangered plant. And she said, can you tell me the exact location? I would like to use it in the USDA research for the management plan. And of course I said, yeah, I'll do that for you. And there have been a couple other examples. One actually that happened last week that was really cool. I had a picture that I had uploaded of it was a Japanese lady beetle, you know, and I can't remember the name of this thing, but it had, the person that saw it and confirmed the ID, they actually recognized that it had a parasitic fungus and said, hey, if you haven't already, duplicate this observation and mark the fungus as the other observation. So sometimes you get more observations than you think you are when you're out in the field because somebody on the platform recognizes there's an issue. I had another one where it was a trillium in the Great Smoky Mountains National Park, and the trillium had a disease, a fungal disease, and a researcher was just looking through photos of trillium to try to find more examples of that disease that they were studying. And same thing, they said, it looks like it has this thing that I don't remember. Can you duplicate the observation? So lots of, this is, it's not hypothetical, but researchers are really using this.
Absolutely.
And I actually heard too, and I need to go find this article, but there have been new species that have been discovered and described that were observed by just citizen scientists that were out there observing things. So you never know what you might find.
Yeah.
All right, let's go back, see if we can't help a couple others here, see what else we can observe or identify, rather. If you guys have any questions, stick them in the comments for us. I'd love to. And actually, if you have something that you've observed that you don't know what it is, drop the link for me. I can pull it up on the screen here and ask Paul-Erik in real time. Okay, northern cricket frog. This is an interesting one. I haven't ever seen this one. Oh, look at how tiny he is.
Yeah.
Oh, and he's still tiny. You may not be able to actually even see that, Paul-Erik, but do you know this species? I see that there's a star here, which means the star means that it is endemic to this area, or it's endemic at least to the United States. So which means that it's native here and occurs nowhere else. It's pretty interesting. Let me see if I can actually zoom any more on my browser. No, it won't let me zoom. Well, here, get out of that. There's not a lot of information. So this is actually a good example of one where there may not be enough visual information to get to an ID, and that's also.
Yeah, it's showing up about the size of a postage stamp on my screen.
Yeah.
But I suspect that might actually be a chorus frog rather than a cricket frog. Not that cricket frogs haven't started calling, they have, but chorus frogs in this area tend to start calling. They're one of the earliest frogs to start calling and breeding. And since that's a very recent metamorph, that one has just recently crawled up on land and reabsorbed its tail. I suspect that that's probably a chorus frog, and it seems to me it's kind of hard to tell from my screen, but it looks like there's a little bit of a dark band going across eyes and down the sides. Are you seeing that?
It does look like that, yeah.
Yeah, so I suspect that that's probably like Pseudacris triceriata. But that's what's great about iNaturalist. Somebody will come along at some point and confirm that. So,
and it looks like that was Corey Hagen that was suggesting cricket frog. So,
well, and that's not out of the realm of possibility. I can't see it very well on my screen, but yeah, I'm inclined to think maybe chorus frog.
Yeah, makes sense. I actually heard the chorus frogs in January this year.
Yeah, isn't that crazy?
It is. All right, how are you on your salamanders?
I'm okay. Yeah, we'll try.
All right, let's try this northern slimy salamander. It looks like there are 5 photos. I don't know if you saw that before I clicked it, but there's a little indicator of how many photos there are. Now, this is one of those things that can really help an identifier. If you're out there observing, take as many as you can get. I mean, a lot of times you're getting a picture as something is running away from you, which is legitimate. And I've put up some really bad bird photos as an example where I'm curious what it is. I'm not necessarily confident anybody can tell what it is, but it's always interesting to see. But if you actually do have something that's relatively stationary, take a few photos of different angles. You don't necessarily know what the diagnostic characteristics are, or in other words, the things that you can see that will help somebody identify. So let's run through a few of these and see if you can see enough to see what's going on.
Yep. And just to add to that, I think that there is some value in putting some kind of blurry photos on iNaturalist, because all of that works to improve the algorithm.
That's true.
A lot of people wind up getting blurry photos, and so if that algorithm can figure out identifications from blurry photos, that just makes it even better.
Great
So
point.
yeah, Plethodon glutinosus. I agree with that identification.
Cool. I'm not. Okay, so here's one thing I didn't mention last time is if you don't have to change the ID and you just agree with it, you can just hit the agree button. I'll go ahead and put life stage and it. Oops, I didn't mean to do that. Oh yeah, well, might as well show you the compare feature since I accidentally clicked it. One of the things you can do if you're trying to just kind of visually identify, and if you're not really sure, you can pull up that compare feature and you can actually you can change the source for this. So you can do RG observations, which means other research grade observations. You can do captive observations, which could be interesting in the example I gave earlier. You can also do misidentifications, which is fun because a lot of times I will, and there aren't any on this one, but I'll see something where I'm thinking, that kind of looks like that, but it seems like something else, and I can't really place what the other thing is either. You can go over to misidentifications, and it'll actually find things that are commonly overridden by the community. So if I say this is X, and then the community constantly comes in and says, no, it's Y, it's Y, it's Y, then that's another thing that is stored in this whole algorithm, and so it knows the things that people are likely to misidentify it as. So that's just really fun. And for me, this is where I get way, way down a black hole because I'll see this northern zigzag and think, well, that's really interesting. And I'll click this link and I'll start reading about that thing. And I'll end up an hour later, through my whole lunch break, like, okay, I need to stop learning about salamanders right now. And then one of the things I'll point out too, you can see the project here. It's automatically added to the City Nature Challenge project for Chattanooga because our project is what's called a collection project. Everything just goes up to it automatically. It's also part of the City Nature Challenge 2020 for the whole world. So that's a fun one to go play around with as well. All right, so I agreed, right? Okay, I'm gonna go back. Anybody has any IDs? Nobody's dropped any IDs for me in the comments. We gotta have some IDs that you need help with. Send them to us. Oh, this is one of my favorites. These little guys. Cope's gray tree frogs, super cute. You'll see these around the greenhouse a lot here at Reflection Riding. We also get them around the animal exhibit. And this person, okay. So there's a comment here that says the color's weird due to the flash, but pretty sure it's a gray tree frog. The description can be really helpful, especially if, let's say, well, plants is probably the easiest example that comes to mind to me. If you take a picture of a plant in sight and you're not ripping it up, it's likely to have a bunch of other plants around it. So, center your photo of the plant, but sometimes it's easier to just go ahead and describe that too, to say, hey, I'm talking about the yellow flower here, in the middle. Let's see if you can see enough. That one's pretty small still too.
Yeah, it's definitely a Cope's gray tree frog. Yeah, I agree with that. Cool. And the comment on the color thing, they can change their color somewhat based on mood. So you can even find Cope's gray tree frogs that are green. So they can be real light or pretty dark or kind of greenish, and it's kind of based on their mood. So yeah, Cope's gray tree frog for sure.
Very cool. We found one over at the greenhouse. It was about the size of a quarter, which is actually smaller, and it was one of the smallest ones I've seen. It's so cute.
That's great.
Oh, here's a good one that I didn't even think about. You don't have to upload photography. Now, if you're using the iPhone app, it only accepts photography. But if you're able to capture sound, which a lot of times for birds especially is a lot easier to get sound than it is to actually find the bird and get a picture of it. You can go to your desktop app and click this little upload button up here, and then you can go navigate to the recording and dump it in. So let me see. I don't know if this will play through the speakers, but I'll try. No, it's not going to play through the speakers for us. But if you're at home and you want to try this, go play that and see if you agree that that's a spring peeper. I will say too, a sound is a heck of a lot easier way to get a spring peeper.
Absolutely.
I've still never actually been able to get a picture of one. All right, so another American toad. Let's see. if you buy this one, let's check the warts here. There we go.
Boy,
that's
a tougher one.
Let me switch to the other photo here. Oh, here you go. That's a little easier to see. So I've got 4 and I've got 5.
Yeah, I think that's another Fowler's toad.
Okay, done. Okay, now other than just blaming it on you, one of the things that it's asking for here, it's saying suggest an identification, then it says tell us why. Mm-hmm. And so I'm going to get you to walk me through this. So I'm seeing 3 and 4, did we say?
Yeah.
And 5 and 6, actually.
So what I would say is 3+ warts per spot equals Fowler's toad. 1 or 2 warts per spot equals American toad.
Cool. And then if I had any other identifying characteristics, what was the other one that you mentioned about?
It's proximity of the cranial crest. You see those crests?
Is that what this is?
Yeah, it's proximity of those to the parotid glands, those poison-filled glands. And I'm trying to remember exactly which is which. But there's graphics that you can look at online. So if you Google American toad versus Fowler's toad, you can sometimes find images that will show you. So I'm looking on my computer right here.
That's actually something that I end up doing a lot is this versus this identification on Google, and there's a lot of good resources out there.
Yeah, so with American toads, the cranial crests tend to be separated from the parotid glands. Like you'll have a fairly large gap relative to what you have with Fowler's toads. Whereas Fowler's toads, they tend to have those cranial crests almost right up against the parotid gland, sometimes touching. But it should be noted too that there's variation in both of these species. And so while these are good identifying characteristics, for most things, it's good if you have more than one thing you can use to identify it. So, it's not totally impossible that an American toad could have more than 2 or 3 warts per spot. And it's not totally impossible that Fowler's toads couldn't have just 1 or 2 warts in each spot. So it's kind of a tricky game, but it's a fairly reliable characteristic, I think.
Okay. All right, let's check out the reptiles.
All right.
Oh, this one's already been identified by a few people, so we can see that community taxon will look a little bit different. There's a black king snake, little early. I've seen 1, I think, so far this year, but these will start coming out quite a bit here as it gets warmer. And this is one of my favorites.
Nice.
It's the beautiful snake, perfectly harmless. Lucky if you get to see one. And this is actually a good way to see when you observe something that a lot of people follow, or a lot of people are interested in, you'll see the IDs kind of stack up a lot of times really quickly. Plants are probably fungi are probably the hardest to identify, and the ones that don't get identified very quickly. And in fact, a lot of them can't be identified until you get into a lab anyways, or at least down to species. But snakes tend to get identified pretty quickly. Birds are like 30 seconds to a minute. Most times somebody will come in and say what it is. Let's go see what else is in here. Okay. See, Betsy's got a couple shells that she found, which is kind of fun. Let's see what else we got. All right, here's one where it says order squamata.
Yeah.
Let's see if we can help. Oh, yeah, cool. Let's see what one of these is better than the other. Maybe this one will help you.
Oh, yeah. Okay, so that what we're looking at there is eastern fence lizard. So that's Scoloporus undulatus. And the only reason I'm so confident about that is because that's the only member of that genus that we have in this area, and that whole group is fairly identifiable. They're the spiny lizards. So if you compared the texture of its skin to, say, a skink or even a green anole, it's much more like armored and spiny, has a rough texture. So the scales are very keeled. So yeah, that would be eastern fence lizard.
Cool. I've seen a lot of fence lizards out west. I think there are a lot more species out west, right?
Oh yeah, lizard species for sure. We have them on salamanders, and they've got us on lizards and snakes.
Makes sense. Cool. All right, let's see if we can get anywhere else here. You guys, let us know. We've got 12 people watching right now. Somebody has to have a species they need help with. Drop a comment for us. Also, just let us know if you have any other questions for Paul-Erik, and if you have anything else you want us to cover. We've got about 15 minutes left on our calendar right here. Oh, and this is a fun one. Actually, this is one I got outside my house. This is a very common, not native, but introduced. And so I'll show you real quick. You see that little exclamation point with the pink thing. This says that it introduced, the Mediterranean gecko was introduced in Chattanooga, Tennessee, and it arrived in the region via anthropogenic means. In other words, people did it. But I just thought this was funny because I walked out the door and he hid between this little tiny thing, and he's like, "You can't see me." But those are pretty common around here. All right, let's see if there's anything else we can help with. So here's a casual observation of a skink. This is somebody that just said, "I saw a skink." There's no photo, but they saw it run by and wanted to record it. Oh yeah. Let me see if there's anything else, or I'll come back to mine real quick. Well, so you mentioned skinks. Mm-hmm. So we might as well jump into the plestodon controversy, I guess you could say. I see a lot of overconfidence with IDs, and let's see if, yep, so somebody says this is a 5-striped blue-tailed skink. Can you tell us why this may or may not be that?
Well, we're not in range for that species, for one thing. Unfortunately with skinks, it's pretty tricky. You kind of have to get some very up close photos of both the head. So you need photos of the jaw, because the number of, we call them labial scales on the upper jaw can help narrow it down. But even that isn't enough to narrow it all the way down, because there's common 5-line skink and the southeastern 5-line skink, which the only way to tell them apart is the scales on the underside of the tail. So skinks are pretty tricky. What you can pretty confidently do in this area is you can tell, at least within this genus Plestiodon, you can tell broad-headed skinks from 5-line skinks and southeastern 5-line skinks by the number of labial scales. But you have to have an up-close, clear photo of the head to do that. And then the other skink species that we have around here is a different genus, Skinkella lateralis, a little brown skink. So Plestiodon fasciatus, yeah, the common 5-line skink, that's a reasonable identification. But as far as confirming that without a shadow of a doubt, not necessarily, because it very well could be either of those other two species that I mentioned.
I'm going to do something that normally wouldn't be helpful, which is I'm going back out to the genus level. I am willing to say this can't be identified.
Yeah, it can't be. That's true.
This photo. So now we're going to have all kinds of disagreement on this page. We'll see what the rest of the scientific community thinks, but I think we got a good answer here. And I actually have no idea why it won't. Oh, there it goes. All right. It wasn't taking my ID for some reason. All right. That was a fun one. Let's go back and see if there's anything else. The other skink that's common in this area is the broadhead skink.
Mm-hmm.
It's probably worth showing people just because you're likely to find it. This seems like a pretty good photo here. I'm going to load that in the background and keep looking. Oh, I got a gray rat snake. We can definitely help with that. So let's click that one, and then I'm going to go show you the broadhead. The internet, I think we're blowing up the internet here as we're all working from home. It's going a little slow. All right, so here's one that you're very likely to see in the Chattanooga area.
Yeah, and that's pretty typical. Distinctive looking lizard as an adult, but again, when they're young, they have bright blue tails and those 5 stripes down the back, just like our 5-line skinks. And so that's why when they're younger, you know, you gotta have some very clear diagnostic photos of scales. But yeah, that's a typical looking adult broadhead skink for sure.
Hey, so one of these webinars that I want to do at some point is what's in a Latin name. Mm-hmm. And I'm curious if you know what Plestiodon is telling us from a Latin perspective.
Plestiodon, I think, has something to do with teeth, but I don't remember offhand. There's actually a really great website that I stumbled across a few years ago that has most North American herp species listed, and it explains what the Latin name means.
Oh, cool.
So I can go and try to find that and get an answer for you later, but I don't remember offhand. That genus is relatively new for this group. So it used to be Eumeces, and I believe that changed in the early 2000s. If I'm right.
One of the things that I love, and again, I was talking about just getting lost, learning about things. You can click all the way back up the taxonomic tree and go learn about things. You can click to this about section. It pulls from Wikipedia, you know, but it also has more information from things like GBIF and the depending on what you're looking at. There may be plenty of good things to click off and check on. But one thing I was going to go do real quick, though, is show you the broadhead skink. You were talking about the broadhead skink as a juvenile. Well, one of the ways that you can go find that is go to view more on the photos, and we can actually just filter down to juveniles. So if you click life stage here, change it to juvenile, and when I was showing you those annotations, this is where those annotations come in as helpful. And boom, right there, you got it. So looks a lot more like that 5 stripes. And that's one of the fun things about this. The other thing you can do is you can see if it is a species that reproduces at a certain time of year, you can actually see, or in a lot of cases, if it blooms at a certain time, you'll see this seasonality curve really sharply. And of course, we're at what, May 1 today. So it looks like we're in the high season here for seeing these around the world. Now, if you want, you could also filter by place. So I'm going to go and just check on when people see these in Chattanooga, see if there's any difference. For whatever reason, Chattanooga shows up at the very bottom after Chattanooga, Colorado, and a bunch of other places I've never heard of. So we actually only have 2 observations, which is surprising. And they've both been in April. So y'all go try to find some.
Well, is that curve based on research grade identifications?
It's both. So if you see this the green curve is the research grade, and the verifiable, so the ones that could become research grade, are up top.
Yeah. Okay. Well, and so again, I think probably the reason there's not very many identifications there is because it really does require up close photos of the side of the head.
That's a good point, and it's actually a good argument to go back up to the genus level and to try it again.
Yeah.
So it looks like we're going to end up with 51 observations at the genus level, and a bit more uneven here, but all the way through the summer, it looks like, into early fall. There's still a lot, though. When you see a difference between the verifiable and the research grade, that means we need your help identifying. So this is a good one that could be useful. The other thing that you could help with is annotations. It looks like the vast majority of the observations have not been annotated, so there are only 2, and then 1 over here. So there are 3 out of these 51 observations that have been described as either adults or juveniles. So, next time you're on a conference call or something with your boss, go start clicking, go annotate some stuff for us. All right, let's go back to that gray rat snake. This is a fun one. It looks like a juvenile here. That's as close as I can get. Hopefully you can see that.
Yep. Yeah, I agree. Pantherophis phalloides, gray rat snake.
Talk to me about rat snakes. There are a few different rat snakes in this area, right?
Well, so yes, technically there is. So we have the gray rat snake and then the red rat snake, which is just another common name for Pantherophis guttatus, which is the corn snake. So corn snake, red rat snake, interchangeable, and that's really why it's best to rely on the Latin names, because common names can be confusing and misleading. For instance, technically, the accepted common name for the rat snake that we have here is the gray rat snake, but as adults, you can very frequently find them around here, and they'll be almost totally black. And so one of the common mistakes that people make is identifying a rat snake that they found as a black rat snake, because it is black and it is a rat snake. So they're not wrong, except that when you call it a black rat snake, it pops up as something else, a different species that we're not actually in range for. So yeah, gray rat snake is our typical rat snake that we have, and then we also have corn snakes, which is a type of rat snake as well.
Here's actually, I did find a corn snake. These aren't necessarily easy to find. Actually, fairly difficult to find in the wild around here.
Yep.
But another beautiful snake.
Yeah, very.
All right, guys, we're getting towards the end of the hour here, and I don't have any more questions. If you guys do have questions, Paul-Erik, thanks so much for coming. If you have questions, drop them in the comments. We'll try to watch the comments for a couple days after this also, and answer any of the questions you might have. If you are on YouTube, go look for somewhere should be a subscribe button. Subscribe to our channel. We are starting to produce more and more content, and we'll keep this coming. And hopefully you can join us in person soon. A lot of these things that we're doing here online, we would normally be doing here on site. And we hope to be able to return to doing that as soon as we possibly can. Paul-Erik, thanks so much for spending time with us. And we hope to welcome you and your biology classes from Hamilton County Department of Education back on the property pretty soon as well.
Thanks, Mark. It's been a blast.
All right. Have a good day, guys.